Okay, couple of things that I wanted to cover before we get started today. So one, some of you may have heard through the grapevine. I'm not through grading all the exams yet, but I'm close enough to being done to know that this one, which I knew ahead of time, was a tough one. I knew that going into it. Okay. You can deem it fair, you can deem it not fair based on how it was. That's your opinion. I would say that it was fair in the sense that the objectives were clearly represented in there. Probably what seems maybe unfair was the amount of points that were on the molecular section, given that that was something we had covered a couple of weeks before that exam even came up. So, given that, what I'm going to be doing is giving these back to you with a raw score on the test on Friday. You get the weekend, you get till Tuesday to make corrections for partial credit on it. So what you're going to see on there are not the answers per se, but just comments that may help you with certain ones. Of course, most of the points can probably be regained on the essay questions, but I'll make anything fair game for it. Okay. And I haven't figured out exactly how I'm going to rescore it. I'll probably put more emphasis on the in terms of points on the essay questions instead of just saying everything's half credit now. And so I'm going to spend some time to think about that. I'll grade them over Thanksgiving break and then give them back to you guys by then. But my goal between today and Friday morning is to at least get the scores in as they sit right now. So that you have that, you know where it stands, and then you can decide if you want to take the opportunity to redo parts of it or not. Okay, and I'll talk more about that on Friday in terms of how I want you to make those changes. So that's one. Any questions about that? Okay. Two. Thanks for answering for everybody. Two. It seems like things are going okay in lab. I'm getting the impression things are kind of running smoothly now. I know I talked to a couple of groups and they had said things went much better this time than last time, and that's kind of to be expected with a project like this. So keep up the good work with that. I think the most important thing is that as you're looking at the results from this first time, making sure you've just taken some time to make any modifications you need on those experiments. In terms of what you've got coming up in lab, we talked about the progress report that's due next week, but in addition, just to note, you've got today, Friday, and then there's still that following Wednesday after Thanksgiving, which is designed just as a day to run experiments. So you have an extra built-in day there. I always build in a screw-up day because it's likely that one of the experiments is not going to work out as planned. Okay. Third. Raise of hands. Who walked away from the mini lecture understanding everything about B cells, T cells, etc.? I sense by the laughter that is not true. Who has a little bit of an understanding? Somewhere between 100% and 10%. Okay. Who walked away going, holy, thanks. All right. We're going to slow down. I want to take today to kind of unpackage that mini lecture some more. So here's what I'm willing to do. I did say that there's a point at which we're supposed to do a couple of lectures of epidemiology. I want to just right now say let's cancel that. We'll continue to focus on immunology during that period. And so that's going to give us a little bit more time to kind of unpackage this. Today I think we'll get through NK cells. Friday we'll do B cells and antibodies, and then when we get back from break and after you guys have done your lectures, we'll come back and we'll cover some more pathogenicity factors and maybe even have a day just to kind of do essay style questions is what I'm imagining right now. With that said, no homework for Friday. Yes. Don't go ahead in the lecture, just...
Wait, hold up, I have the summary thing for Friday.
So summarize what we talk about in class on Friday. So you summarize what we talk about in class. B cells and antibodies. Good? Okay, Danica.
said this, but for when we're turning in the topic and objectives for our group project, we do that on Blackboard.
Yeah, so what I have done, maybe I should just go to this real quick. So the due date for them technically is end of class time on Monday. Okay, but if you have it done before class time on Monday, and if you've got your topic picked before class time on Monday and have it posted, I'll throw on an extra credit point on top of it. Okay, so basically, do a little work over the weekend and you get a freebie. Haley.
For the objectives, are we supposed to like make up our own objectives that are like our topic, or do we pick from the objectives?
You pick your own, and you only have to come up with one. I think going more than one may be a little tough given the limitation in time. So I think one objective is reasonable for this. Okay. And then the questions that you develop should really be a way to measure, can they actually achieve those objectives? Okay. And this is stuff that I can work on with groups. That's kind of why we have this time set aside Monday to work on this. Nicole.
This is kind of off topic, but do we have any homework over Thanksgiving?
Your homework over Thanksgiving is to put the presentation together. So, and this should be, I think VoiceThread is a good way to do this one because everybody's going to be at separate locations. So what I would say is Monday next week, probably what you want to do Monday is take the time to decide. I imagine these are going to be 3, maybe 4 slides at most. That's it. So what's everybody's role in that? You need to have somebody that comes up with the questions and probably puts together the answers as well. That's the homework over break. Okay.
So it is group. It's not individual.
Oh yes, this is so the groups that we were kind of assigning over the last few days, those are the ones. Now, if you were one that like didn't give me a group or something like that, I threw a group together. So let me know, but everybody should have a group now for it. Yeah, so we have Monday to work on it.
I thought this was individually.
No, this is group, and then I'm around all day Tuesday, though there's nothing scheduled, so you can come and bother me on Tuesday if you want to as well with questions. We good now? Allie, no, apparently not. Go.
This isn't about this. So, for the corrections, you want us to hand them into you on Tuesday?
Yes. Next week, Tuesday.
Okay.
Can we do that Monday?
Sure. Sure. By Tuesday is basically the deadline. By the time I leave my office on Tuesday.
We're just writing on a separate sheet.
I'll talk about that on Friday, but that's what I imagine is that I'll have you hand in your original and the modified one where just the answers are either hand typed or on loose leaf paper. Yeah. Yes. But you can hand it in Monday if you want. Go for it. I'm just giving you to Tuesday because some people don't want to do it over the weekend. Jasmine.
Can you just like give me an example of like what you do for a project?
One of the groups is so one of the groups from the morning class wants to talk about extremophiles. So microorganisms that live in extreme environments. So that's their broad topic. I don't know if they're going to focus on 1 or 2, but that's kind of where they're going. Examples. Where are you going with yours? Did you decide yet? Sorry, Matt and I talked about this yesterday, so.
I'm looking into it. One thing I was thinking about was like probiotic delivery.
Okay, so probiotic delivery. How's it being done? Okay. One could be, gosh, I mean, there's so many options. Destruction of biofilms. It could be something medically relevant. It could be something not relevant to that at all. Chlamydia and rickettsia. You want to talk about chlamydia for 5 minutes? Talk about chlamydia for 5 minutes. So Jasmine, honestly, I do not care what the topic is. It's just got to be related to microbiology. Okay?
Should we do it like it's our own mini lecture?
That's how I want you to do it. Yep.
Like, is there like 15 minutes?
Sorry, they've gotten a little long lately. I apologize for that. I apologize. There's a lot of information, so I apologize for that. Danica.
You said you were going to copy and paste the objectives so we could study from. Are we being tested on what everyone
Ah, yeah. So Danica asked a good question. So are you being tested on what everybody else is doing? Yep, that's part of the final.
Wait.
Oh,
That's part of the final, or that's the whole final?
Oh no, it's part of the final. So right now, how I am envisioning this is that the multiple choice and fill in the blank for the final is going to come from the mini lectures.
And then the other half is what we're learning right now.
Part and vaccination.
Wait, is it not cumulative?
So the final is 100 points are new, 50 points are cumulative. It's in the syllabus, been there since day one, guys. Jenna.
So
if you make a voice thread for your presentation, will you just play the voice thread?
Yeah, that's what I'm going to do. So I want you guys to make voice threads, share them with me, and then all I'm going to do is at random pick the next one up, and we're going to play it.
But like, so we're not presenting in front of
No, you're not physically presenting the lecture. You are videotaping a mini lecture, essentially. Hey, wait, now I can't hear a damn thing, guys. Okay, go ahead. Yeah, mini lecture, and then you got to stand up here for about 2 to 3 minutes and answer any additional questions.
Can we like leave the room while you play it?
Nope. No, I'm going to force you guys to stand here and listen to it, which I don't do myself, but I'm going to force you to do it. Yeah.
When it means under organization, it says like narrators always use cues such as pointing. Does that just mean like with arrows on voice thread?
Yeah, or like saying, so like in the image to the right.
Like an actual video of us. It's just like talking.
Yeah. Yeah.
I don't really want people to watch.
No, it's going to happen. Sorry. I don't like people watching me either, but guess what? It happens 2 times, 3 days a week. All right. Let's talk immunology. All right. Here's where I want to start. Let's review the steps of phagocytosis, because this actually becomes really important when we talk about how do we move from the innate to the adaptive immune response. So what's first in phagocytosis? Attachment. Between what? PRR on the cell and a PAMP on the pathogen. Okay? After that, we get the phagosome to form, matures, phagosome fuses with lysosome, and then what happens? What's the last step of this process? Death, destruction of the pathogen. And that's where the adaptive immune system begins to pick up. So what we're going to focus on first is what happens to the pieces of that pathogen afterward. Now, in the mini lecture, I use the term antigen a lot, which essentially what that term means is it's antibody generated. So an antigen is any molecule, typically a protein, that would trigger B cells to produce an antibody. In other words, it's something that activates the adaptive immune response. Allie?
Where are antigens?
Great question. So an antigen oftentimes is found on a pathogen. So like an outer membrane protein on E. coli, that is an antigen. It's not as if you can look at something, point at it, and say, yep, there's an antigen. Basically, it's anything foreign inside the host cell that activates an immune response. So in terms of E. coli, with the 90 or so different outer membrane proteins they have, each of those is a potential antigen. Okay. The other term we sometimes use is epitope. And an epitope is a piece of an antigen. So what the adaptive immune response, I'll use the terms interchangeably probably, but typically if we're talking in strict immunological terms here, what the adaptive immune response recognizes are pieces of an antigen or an epitope. So if a protein is 200 amino acids large, that's the antigen. Epitopes are usually on the size of about 10 to 20 amino acids total. Okay? So, given that, out of a 200 amino acid protein, you got somewhere around 10 to 20 different epitopes that could be produced. Right? And what we're going to talk about is, one, how those epitopes get loaded into what we call MHC1 and MHC2. And then how does the T cell respond to that? How do T cells become activated? All right. So let's start with the basics. Oh, bugger. All right, so MHC1 and MHC2 stand for Major Histocompatibility Complex. You can call them MHCs. They are probably the biggest difference between the two of them is what kind of antigens do they load into this MHC. And the MHC you can kind of think of, oftentimes it's kind of drawn like a stick to show that it's a protein embedded in the membrane, and then something like goalposts or a half circle or something to say that it's cradling an epitope in it. And it's essentially presenting this epitope on the outside surface of the macrophage. So two different types of MHCs. MHC2, we'll go through first here. MHC2 is going to present antigens that come from outside of the cell and then are brought into the cell by phagocytosis. In other words, as a microorganism binds to the surface of a macrophage, binding between the PRR and the PAMP, and that pathogen gets brought into the cell, gets brought into a phagolysosome, gets broken down into small little pieces. Eventually what we find is that within those phagolysosomes, you'll get another membrane vesicle containing MHC2, and that's going to fuse to this phagolysosome as well. When that happens, those little epitopes or pieces of the pathogen that have been destroyed will individually, one to each MHC2. So get loaded into these MHC complexes. That gets exocytosed so that now MHC2 is on the outside surface. Okay? So again, major points here, MHC2 will present extracellular pathogens that are being brought into the cell and broken down by phagocytes. We only find MHC2 on what we call PAPCs, or professional antigen presenting cells. And we're going to talk about two major types of professional antigen presenting cells. One of the things I've said so far a couple of times is this occurs through the process of phagocytosis. Which cells are capable of phagocytizing pathogens? Macrophages are one, neutrophils, and what else? B cells. Two of those we consider to be professional antigen presenting cells as well. The B cells and the macrophages. Neutrophils do not express MHC2. They have the gene for it, it's in the genome, they just never express the protein. Macrophages and B cells are the only ones that do. We'll come back to the role of this in B cells and their activation on Friday. We'll come back to macrophages a little bit later today. Okay? Go ahead, Allie.
When the MHC2 is on the outside, like the little fork thing, is there like a piece of the pathogen in it?
Yes, thank you. There is. And those pieces could be different. I mean, it's dependent on which proteins or which part of proteins get loaded in there. So you can have a macrophage that has ingested one bacterial cell and might be presenting on its surface thousands of different epitopes, each one a different piece of that bacterial pathogen. Okay, you can already see maybe from that, we'll kind of talk about the interplay later on, but there's an importance of that, taking one pathogen in, presenting multiple epitopes at one time, and amplifying or increasing the immune response. Okay, so that's MHC2. Works. Oh, Nicole, sorry.
What are those letters up at the top?
PAMP, PRR.
Below. Oh, PRR.
Yeah, sorry.
No.
I'm writing with my finger because my pencil is dead. So. Really good mechanism against bacterial and viruses that are kind of floating around in the tissue. The other mechanism of this, can I erase this one? Is that okay with everyone? All right, so the other mechanism we find in all cells that have a nucleus. In other words, the second mechanism for MHC1 occur in all but red blood cells. In MHC1, what we find in these cases is that the pathogen is actually already inside of the cell. So we consider MHC1 oftentimes a process for intracellular pathogens. And these pathogens would be found, for example, in the cytoplasm, like this gigantic virus inside of this host cell. All of our eukaryotic cells have a complex of proteins known as a proteasome. I'll write that term out real quick. And the proteasome, honestly, in a lot of pictures, kind of looks like a garbage can with a lid. This is really what it looks like on a three-dimensional scale, too. The function of the proteasome is to break down proteins. And it's doing this all the time. It's actually an important part of homeostasis because proteins aren't lifelong. They will usually retain their normal function for few hours depending on the protein, but eventually they degrade. And when they degrade or start to break down, they get sent to the proteasome, broken down into individual amino acids, so that those can be recycled to produce new proteins again. And so what the cell is doing here is it's actually kind of always taking these products from the proteasome, breaking them down into smaller pieces, and eventually, and we're not going to go through the exact mechanism by which this one occurs, these will get loaded onto MHC1. And so one of the interesting things about MHC1 is that it actually will present both the cell's own proteins or own pieces of proteins on the surface. So it presents self and it presents non-self. It will present on its surface potential pathogens or pieces of potential pathogens as well. And we're just going to focus on the second of those two examples.
So, what was the structure with like all the little labs?
This would be the virus going through the proteasome and now getting broken down into its individual. Oh, this right here?
Yeah.
Virus. We'll call it human papillomavirus today. So then what's going to happen in this process is that on the surface of these cells, we have MHC1, again presenting all sorts of different epitopes for this pathogen. So, major differences between MHC1 and MHC2. They both load epitopes or pieces of antigen. MHC2 only found on professional antigen presenting cells. MHC1 found on all cells with a nucleus. This becomes important later on. The other major difference, the types of potential pathogens. MHC2, those that are being brought outside into the cell. And then MHC1, those that are typically what we would consider intracellular pathogens. One's like a virus that needs to be inside of a host cell in order to replicate. Right. Any questions on the contrast between those two?
What types of different cells have like the MHC thing? So just is it just non-nucleated?
So you mean MHC2 or MHC1?
Because
all of them within the.
Yep, so MHC2 would only actually macrophages and B cells. So those that we defined as our professional antigen presenting cells. All right, so let's compare these a little bit further. All right, so far we've said MHC1 found on all cells, MHC2 found on professional antigen presenting cells. We'll come back to the importance of that in the next few minutes. But what I want to talk about next is what do they actually interact with? Because somehow this presentation has to now relay a signal that there's an infection occurring, an immune response needs to be activated. So how does that occur? And this starts to then differentiate into the response of MHC and MHC1 and 2. They actually interact with different types of T cells. So one interacts with the T cytotoxic or the TC cells, sometimes called cytotoxic T cells or CTLs. And the other interacts with T helpers. Okay. So let's take a look at that interaction. All T cells, regardless of whether they're T cytotoxic or T helper, have what we call a T cell receptor, or a TCR. Each T cell that is produced has a unique T cell receptor. And we won't go into the specifics of how each unique one gets generated, but the most important thing to note here is that each of those unique ones recognizes a unique epitope, okay, or a unique antigen. The T cytotoxic actually have the T cell receptor and then what we call a co-receptor, and the co-receptor that it has is called CD8. And it's not important what the CD stands for. Oh my gosh. All right. So let's draw a macrophage here. Macrophage. We've already said that the macrophage, or these professional antigen presenting cells, have on their surface MHC1 and are presenting some sort of antigen on the surface. Two things need to happen at the exact same time for a TC to become activated. And in essence, the role of the MHC complexes, or MHC class 1 and MHC class 2, the function of those is to activate these adaptive immune cells, particularly the T cytotoxic and T helpers. So two things have to happen at once. First, T cell receptor has to be able to bind to that epitope. Secondly, CD8, this co-receptor here, has to recognize MHC1. If those two things don't happen, the T cell remains inactive. Okay. This is kind of a way, I mean, if you've ever seen cheesy movies, we've been watching Goonies at our house a lot lately, which is like the best thing on the face of the earth. But if you've ever watched a movie where like two people, both with keys, have to unlock at the same time for something to happen, that's what's happening here. We have to have two events take place simultaneously to unlock or turn on this T cytotoxic cell. And there's a couple of things that happen that lead to that. One of the events here is that when this binding occurs, the T cell begins to change gene expression, but what we also find on the end of the macrophage produces cytokines. Those cytokines activate the cytotoxic T cell. Sid.
What were the other ones that were to happen?
So CD8 has to recognize MHC1, and the T cell receptor has to be able to bind to the epitope. And so in other words, if this cytotoxic T cell is just kind of like floating around and it recognizes the epitope on the pathogen, but it's not in an MHC1 complex, that T cell remains inactive. The only time it happens is when it's first interacting with a macrophage. Alan.
Wait, what does the yellow arrow represent?
Cytokines.
And they have to go to the T cells.
Yeah, so what happens is the macrophage, what will happen is this interaction here actually triggers a series of events inside the cell, ultimately changing the expression of genes. Cytokines get produced and then get secreted from the macrophage and will hit the T cell basically, and then will kind of excite the T cell to go from an inactive to an active state. So actually what's happening here is in some ways, there's 3 stimulatory events that are occurring at the same time. T cell receptor binding to the epitope, CD8 binding to MHC1, cytokines from that macrophage, all happening at once. In the case of MHC2, the T cells are different. It's a different class. And the particular ones here are the T helpers.
Is this for MHC3?
Yes, yeah. So the T helpers interact with the TCR. Or sorry, T helpers have a TCR. In this case, it looks different than the other one, indicating a unique cell. The T cell receptor is interacting with the antigen loaded in MHC2. What else do we have to have? Yeah, so this is, I would have taken anything like a co-stimulatory molecule, but I'll take CD4 definitely. CD4 is the co-stimulatory molecule here, or the co-receptor. And CD4 recognizes specifically MHC2. Where else have we seen CD4? We actually talked about it when we talked viruses. Which virus uses this as a receptor? Yeah, HIV. And so these T helpers are the only cells that have CD4 on their surface. There are some macrophages, but it's mostly T helper cells. And so we'll talk about the implications of that a little bit later, but you can already see where this might be a problem. If HIV is infecting T helper cells, think about what might happen there, and that's something we'll come back to in the next couple of weeks as we continue to talk about this. Same thing happens here as what we observe on the other side. Macrophage secretes cytokines to activate the T helper, and actually in return, T helper secretes cytokines that will activate the macrophage, will activate itself, and can even activate the cytotoxic T cells. So the role of a T helper in probably the most simplified description we can give is the T helper is a regulator of the adaptive immune response. It's kind of like a foreman at a construction site, or think of it like a boss. The individual who is head is telling everybody else what to do. They're the one that's ultimately regulating and controlling the response. And the way that T helpers do this is by secreting cytokines. And the specific cytokines that get secreted is dependent on what kind of infection is occurring. But largely the role here is for those T helpers to improve the activity or enhance the activity of the other cells that are contributing to the immune response. So I'll pause here real quick and just first see any questions on this concept of activating those T cells. More cytokines. Sorry.
For like the little dotted one.
Cytokines.
So it's like
Cytokines. Cytokines. Yada yada. Everywhere. Matt?
Do all the cytokines, are they all produced at the same time, or does one have to be produced first to trigger the others?
Yeah, that's a great question. Individual cells produce different types. So it's more the type of cell that defines the cytokine that's going to be produced. Like macrophages will produce a kind that only stimulates T helper or T cytotoxic. And actually, one of the things, this is getting a bit more complex than we really need to. I mean, in general, cytokine. But the yellows on top here, it would be a different set of cytokines that activate the T cytotoxic cell than those that activate the T helper. And what drives which of these get expressed is really this interaction here, which triggers some sort of change of gene expression inside of the cell. The T helpers secrete a completely different set and actually secrete about 3 or 4 different ones at one time to stimulate macrophages, to stimulate itself, and to stimulate the cytotoxic T cells. So it's kind of simultaneous in the sense of the T helpers. Jasmine.
So the TC cells, like what is it?
We haven't covered that yet. That's actually what we'll do next.
Okay, so the T helper cells activate cytokines.
They secrete cytokines to activate other cells in this response. And we'll revisit that in a minute too. Yeah. So if you're doing word association here, things to kind of keep together. CD8, MHC1, cytotoxic T cell, intracellular pathogens. In the case of MHC2, CD4, T helper, extracellular pathogens, just to kind of keep this straight right now. Paige.
So is CD4 present only on the T helper, or is it also present on the B cells?
It's not present on the B cells, actually.
What
It's-
would it be for that one? Like for those?
It's a different set of proteins known as CD28 and CD3. I'm not going to ask you to memorize those ones. Immunology is a concept that is way over my head sometimes. Okay. And it is a challenge to teach. All right. So Jasmine asked, what do those cytotoxic T cells do? Okay. Here's what they do. Those cytotoxic T cells, once they have been stimulated, they begin to produce inside the cell 2 different enzymes that are kind of stored, or 2 different molecules that are stored in vesicles. Those molecules are known as, oh bugger, I always do this. Someday I'm going to learn. Known as granzymes. And perforin. Perforin perforates. In other words, it pokes holes in the membrane of cells.
Did you say that these T cells secrete that?
Yes, they will, right? So before they secrete it though, Nicole, once they become activated by that macrophage, they just start to produce boatloads of it in vesicles inside the cell.
Yeah, so it has to be activated in order for them to.
Yes, yeah, yeah. And they'll start to produce these in large amounts. Then becomes then we get the secretion process. So how do we get that? Well, we said all cells have an MHC1, right? Including cells that might be infected with a pathogen inside. Those cells present pieces of the pathogen. If a T cell, or if a cytotoxic T cell recognizes that particular epitope, and CD8 binds to the MHC1 complex, what happens is that T cell all of a sudden just releases all of these toxins from those vesicles to this infected cell. And 2 events take place. One, perforates holes in the membrane. And it's still debatable as to what the advantage of this is, but it seems to cause death of that cell. And then the granzymes trigger apoptosis or apoptosis, depending on how you say it. There is a correct way to say this one, but we'll leave that for debate. What is apoptosis/apoptosis? This was like 155 maybe, if you learned about this. Yeah, it's programmed cell death, right? It's basically, sometimes the word that's used is cell suicide. So those two molecules seem to program this infected cell to kill itself. What's the benefit of this? Can't make more what? Or think about what's in there. Replicate the virus. In other words, you've basically ended that lineage of the virus in that cell. That's the goal of this. So cytotoxic T cells, by producing these chemicals, very specifically tell individual cells to die because those cells are presenting something on their surface that isn't normal. Could be a piece of a virus, could be, honestly, could even be a mutated protein. In the cases of cancer, one of the hot topics right now in cancer therapy is cancer immunotherapy. Take somebody's T cells out of their body, train them to recognize the mutated protein or proteins that are on the surface of those cancer cells, and then have them kill them by this mechanism. T helpers, on the other hand, this breaks it down into two different types of T helpers. There's actually 4 different types that we know of right now. I'm not asking you to know the types, but just to know in general, T helpers, depending on the subtype and the different subtypes produce different cytokines. Those cytokines will activate different sets of cells. So we've got some T cells that will activate CTLs, NK cells, macrophages, neutrophils. In other words, increase production of granzymes and perforin or increase phagocytosis. And then there are some that activate B cells. And this is something we'll talk about on Friday, is that activation. Okay. Sarah?
Wait, we talked about like the ones where their own cells die quick again? Just recap.
The cancer or the, oh, okay, so the T cell, those activated cytotoxic T cells are producing granzyme and perforin. If the T cell receptor binds to the epitope, if CD8 binds to MHC1, these T cells secrete that perforin and granzyme, pokes holes in the membrane, programs that cell to die. And again, the benefit of this is if that cell dies, if it's truly infected with a virus, for example, it actually means the end of that virus as well.
So it's killing an infected cell.
It's killing an infected cell, yes. Jasmine. Go over the T helpers? Yeah. So the main role of T helpers, secrete cytokines, and the specific cytokines that those T helpers are secreting will activate NK cells and cytotoxic T cells to produce granzymes and perforin, or will activate macrophages and neutrophils to increase phagocytosis. No, you guys can draw a flow chart though. That's a great idea.
Wait, what? I thought we just went over T cells.
We did.
On the last slide? About, you said the role is to regulate that adaptive immune response.
Yeah. Yeah, I'm telling you specifically how they do it now. For the 3rd time. Secrete cytokines. Activate innate immune cells. Secrete cytokines. Activate B cells. Period. Sid? Okay. So increase phagocytosis. Increase Granzyme and perforin production. Increase B cells, or enhance B cells to produce antibody, and that we'll cover on Friday. So ends the 3rd chapter of Zach, or book 3, chapter 5, verses 1 through 6. Last thing, before we head out of here, 2 things I'd like you to think about. Some viruses prevent MHC1 from ever making it to the surface of the cell. What's going to happen to that cell? Okay? So spend some time thinking about that. Again, some viruses prevent MHC1 from ever making it to the surface of an infected cell. What's going to happen? What does that mean in terms of the viral replication? What does that mean in terms of killing that infected cell? You may want to take a look ahead, 1 slide beyond this, which could actually help to explain a little bit more. All right, so the last thing that I'm going to do is let's just go back to the initial things here. So, what we've covered today. That first one there, that's the final question I'm leaving you with. How does a virus that blocks MHC1, how is that going to affect the immune system? How does that contribute to that organism's pathogenicity? We've talked about the function of T helper and T cells, and T cytotoxic, sorry. 11.5, 11.6, we'll cover these on Friday. Okay? Sarah? Virulence?
That's the thing that blocks MHC1.
Yep, exactly. Okay.
TC cells are the same thing as these are saying T cells, right?
Yes, yes.
What about TH cells?
T helper. So TC cells are cytotoxic T cells. Sometimes they are called CTLs as well. Goes under 2 names. All right. Draw some flow charts for yourself. Maybe a concept map or 2.
Or 7.
Or 7. See you later.